today's talk I'm giving, but I'm going to be talking about the work of a PhD student in my lab, Andrew Best. I'll give the full acknowledgements at the end though. So the subject of my talk today is the role of Tra2 beta in breast cancer. So just to give you a bit of background first about the Tra proteins, or the Tra2 proteins I should say. Tra2 proteins are very conserved splicing regulators. They're found in fruit flies where there's a single Tra2 protein involved in sex determination. And they're also found in mammals as well, where there are two proteins, Tra2 alpha and Tra2 beta. Now, in structure, they have a central RRM flanked by arginine serine-rich regions. So central RRM flanked by RS regions. They're reminiscent of in structure to the core family of SR proteins, but the SR proteins instead have an N-terminal RRM and C-terminal RS domains. So they're a bit different in structure. And another difference is the Tra2 proteins so far have only been implicated in regulated splicing, whereas the SR proteins have also been implicated in constitutive splicing events as well. So we became interested in a possible role of Tra2 proteins in breast cancer following on from a paper from Stefan Sam's lab in cancer research, which showed that Tra2 levels were elevated in invasive breast cancer cells. And we looked ourselves at some data which was available on the web in a database called cBioportal, which showed that in a larger cohort of breast cancer patients that Tra2 was elevated in around the mRNA level in about 10% of tumors. Tra2 alpha, which is the other Tra2 protein in humans, was also elevated in some tumors as well. And in the same cohort, SRSF1, which is a known oncogene from Adrian's work in breast cancer, was also elevated. So the content of my talk, I'm going to be talking to you about human Tra2 proteins. So this is the icon of humans from the UCSC genome browser. And absolutely essential, we found, to understanding what these proteins are doing in breast cancer cells was finding out the relationship between Tra2 alpha and Tra2 beta. So once we'd discovered that, we were able to actually work out exactly what they're doing. So the first thing I'm going to tell you about is what's the relationship between Tra2 alpha and Tra2 beta? So that's my first question I'll try and answer. Now, on this slide, I'm showing three Western blots taken from the same cell extracts. So at the bottom is the loading control, alpha-tubulin. In the middle, the Western blot's probe for Tra2 beta, and the top is probe for Tra2 alpha. Now, these Western blots actually explained a lot to us because we've been trying to raise antibodies against Tra2 alpha for quite a long time, but we always got a very weak signal. And this is shown in the first lane. Tra2 beta gives you a big band. Tra2 alpha gives you a much weaker band, although it does disappear when you siRNA for the Tra2 alpha mRNA. But something very remarkable happens when you do an siRNA against Tra2 beta instead. So if you siRNA against Tra2 beta, the Tra2 beta signal disappears, but the Tra2 alpha signal shoots up. So this told us that actually in this cell line, and all these experiments are carried out in a cell line model of metastatic breast cancer, which is called MDA MB231. In this cell line, the Tra2 alpha protein is negatively regulated by Tra2 beta. So this wasn't a total surprise to us because previous work from our group had shown that Tra2 beta actually binds to a poison exon in the Tra2 alpha mRNA, but the strength of this effect was surprising to us. So this means that the Tra2 proteins are a bit like the PTB proteins. You've got a dominant protein and a non-dominant protein. So the next thing we did, given that Tra2 beta is the dominant protein in the cell line, was to try and work out where Tra2 beta was binding in these cells. And I'm going to show you a picture of that next. We used iCLIP. So this is there's a lot on this slide, so I'm just going to take you through it. What we did was we did iCLIP in 3 biological replica experiments, and we got around 7.5 million reads that we could map back to the human genome, 3.5 million of which are unique reads. And this is showing Tra2 beta binding sites in the Tra2 beta gene and the Tra2 alpha gene. And underneath are RNA sequencing data from the same cell line. So all the Tra2 beta binding sites map to these poison exons in the respective genes. So Tra2 beta binds to its own poison exon, but also binds to the poison exon in the Tra2 alpha gene. Now, if you notice, these sequencing reads taken from the same cell line show peaks over each of the exons, but not above the poison exon. And the reason is because if that poison exon is included, the mRNA is less stable. So even though you get a high number of CLIP tags mapping here, you don't get much stable accumulation of mRNAs, including that exon. Another thing to notice from the sequencing reads is the height of the y-axis. So for Tra2 beta, the y-axis goes up to 1,749, whereas for the Tra2 alpha gene, it only goes up to 476. So Tra2 beta is actually expressed at much higher levels as a gene than Tra2 alpha in the cell line. And that's possibly why it's a dominant protein. So we mapped all these CLIP tags onto the human genome, and we wanted to see what happened to splicing of these the exons when we removed Tra2 beta. So what we predicted was there should be a splicing change. So this is an example of one of our exons. We've looked at a number and they all gave us the same result. So this example exon is in a gene called GlyAL1, which is on chromosome 16. This is the regulated alternative exon with the Tra2 beta iCLIP reads. What happened, this is capillary gel electrophoresis from one experiment. This is 3 independent repeats. What happened when we depleted Tra2 beta was absolutely nothing. So this is exon inclusion. This is exon skipping. So there's very little change in exon inclusion when you deplete Tra2 beta. And likewise, if you deplete Tra2 alpha, there's very little change in splicing. But a very dramatic thing happens if you deplete both Tra2 alpha and Tra2 beta. So then you get significant skipping of this exon. So this tells us that Tra2 alpha, when it becomes upregulated, is able to functionally compensate as well for Tra2 beta. So given we had to do a joint depletion to see significant effects on splicing, we focused our search for looking for Tra2 beta regulated exons by doing joint depletion. And we hoped that this would tell us exactly what Tra2 beta was doing in breast cancer cells, which might be important for them. So this shows the results here. We managed to identify over 50 new targets for Tra2 proteins in this cell line. This plot shows percentage splice changes when both Tra2 proteins are depleted. So at the top is a 0 change, so no effect, and at the bottom is 100% change, which is a total switch. So on average, we saw around 25% splicing change, although some exons were very small changes, and incidentally, this included SMN2, whereas other exons had very significant changes, and all the exons which changed more than the average I've highlighted in red here. Now, first of all, I'm going to tell you what these exons tell us as a collection about what Tra2 proteins are doing in this cell line. But then I'm going to focus on 2 specific exons which are of interest. So first of all, what are these exons telling us as a whole? We looked at the genes in which they resided, and we did gene ontology analysis. And what we found was significant enrichment for particular terms, chromosome organization and response to DNA damage stimulus. Now, these particular Go terms are somewhat similar to each other, and so some genes actually mapped within both these 2 different Go terms. Now, this accounted for a proportion of the genes, but we also mapped all our genes onto the BioGrid network which looks at protein interactions, and we found that a further 61% of our genes which didn't map within these Go terms actually functionally interacted with a protein which was a member of one of these Go terms. So it seems there's a significant enrichment in Tra2 function in chromosome organization and response to DNA damage. Now, I'm going to tell you about two specific exons here. One of them is an exon in CHEK1, and the second one is an exon in a gene called ANKRD1. So first of all, CHEK1. So this shows you RNA sequencing data and CLIP data for the CHEK1 gene. This is the CHEK1 gene here at the bottom on the UCSC genome browser. These are the sequencing reads for control cells and double knockdown. And you see this exon changes when you do double knockdown of Tra2 proteins. And this is the alternative exon at the 5' end of CHEK1. And this maps to a huge density of iCLIP reads. Now, Ben was talking yesterday about the properties of alternative exons frequently being between protein domains. In fact, this exon in CHEK1 is an exception because this is actually an exon which is situated within an absolutely critical domain. CHEK1 encodes a serine threonine kinase, and this exon encodes part of the kinase domain, so it's an important exon. Just to let you know what CHEK1 does, it's called CHEK1 because it's a checkpoint regulator, and it actually regulates two specific time points in the cell cycle. This is the cell cycle, so what CHEK1 does, it regulates the transition between G1 and S by phosphorylating a protein called CDC25A, and that leads to degradation of CD25A by the proteasome. And it also regulates the transition between G2 and M by phosphorylating another protein called CDC25C. So what happens when you disrupt the splice? So these are protein extracts taken after double depletion of Tra2 proteins, 72 hours after depletion. Now, if you do a Western blot of control cells or double knockdown cells, and you probe with an antibody which recognizes CHEK1, you can see two bands in the control. This is plus the kinase domain, and this is minus the kinase domain. So you get two protein isoforms as well as two mRNA isoforms. But after double knockdown, you get a switch to the shorter isoform only, and you lose the longer isoform. So you remove the kinase domain from CHEK1. And I mentioned CDC25A is one of the phosphorylation substrates of CHEK1. And when CDC25A is phosphorylated, it becomes destabilized. So exactly corresponding with the loss of the kinase domain, we see stabilization of CDC25A. And this is the actin loading control. So this is 72 hours after depletion. 72 hours after depletion, the cells look okay under the microscope in terms of proliferation. But if you look 96 hours, you start to see significant changes. So these are control siRNA treated cells. This is a FACS profile to show the cell cycle distribution. This is G1. This is G2, and in between you've got cells in S phase. So most of the cells are in G1, but if you double deplete for both Tra2 proteins, you get a decrease in the number of cells in G1, much more cells in S, and more cells in G2. So that is consistent with there being a cell cycle defect caused by the loss of the CHEK1 protein. Now, normally we think of checkpoints as being things which are overridden in cancer cells. But in fact, the checkpoints which are controlled by CHEK1 are very important for cell viability because you don't want to go into mitosis before you're ready, and you don't want to go into S phase before you're ready. So you'd think if you're missing these checkpoints, it would actually be harmful to the cell. And in fact, when we measured cell proliferation 120 hours after we did the depletion, that's what we saw. These are untreated cells. get lots of proliferation. These are cells treated with control siRNA. Still lots of proliferation, but if we double deplete for Tra2 proteins, we decrease cell proliferation. So this is an important checkpoint in these cells. And interestingly, if you just single deplete either Tra2 beta or Tra2 alpha, you see no effect on cell proliferation, which again shows that these two proteins can compensate for each other. Now, the second exon I want to tell you about is an exon in a gene called ANKRD1. Now, this exon was a total surprise to us because Tra2 proteins have previously only been implicated in alternative splicing, but in fact, this exon in ANKRD1 is a constitutive exon. So this means that Tra2 beta, as well as being involved in regulated splicing, also plays a role in constitutive splicing. And this data is shown here. This is the regulated exon here. So this is RNAseg from controlled depleted cells. This is exon 3. And you can see there's quite a high peak. This is after double depletion for Tra2 proteins. So this peak diminishes. So this is one of our high-frequency splicing changes. And this is not annotated on the UCSC genome browser as an alternative exon. And we find no evidence ourselves for it being an alternative exon. So it seems to be constitutively included. So that means that broadens the role now of Tra2 proteins. They're also involved not just in splicing regulation, but in constitutive exon inclusion as well. And since this was an unexpected result, we actually made a mini gene containing this exon, and we looked to see where the Tra2 sites might be, and then we mutated them. And I'm not showing you the actual mini gene itself, but just to summarize what we found, there were 2 clusters of Tra2 binding sites. If we mutated either one cluster or the other cluster, we affected splicing of this exon. So it seems to be a direct target, not just based on CLIP analysis, but by mutagenesis as well. So do other RNA binding proteins affect these exons? This is the last piece of data I'm going to show you. We looked at these 2 exons in the 231 cells, which were either treated with the control siRNA or were depleted for one of 53 different RNA binding proteins. Now, this plot you notice a negative number. If there is a bar here, it means there is an inhibition of splicing. So if for ANKRD1, which is a constitutive exon, if you deplete any one of 53 RNA binding proteins, the only combination that we see that has an effect is depletion of Tra2 alpha and Tra2 beta together. So this exon, this constitutive exon, seems to be uniquely dependent on these proteins, whereas CHEK1, which was the alternative exon, is also affected by Tra2 alpha, Tra2 beta depletion, but it's also affected by some other RNA binding proteins, interestingly, including some components of U2 SNRP. So just to summarize the data, this is our model of what's happening in the cell line. In control cells, Tra2 beta binds to exons, and one of the exons is a poison exon in Tra2 alpha, and this represses Tra2 alpha expression, but it gives you good inclusion of exons which are important for cell growth. If you deplete Tra2 beta by itself, you get upregulation of Tra2 alpha, and that can maintain cell growth. But if you do a joint depletion, then you start losing exons, which are important for cell proliferation. And under these circumstances, you lose cell growth. Now, just to give my acknowledgements finally, I would like to thank the Breast Cancer Campaign who funded this project. Also, the other groups, the other organisations that fund my research, the BBSRC, Wellcome Trust and Prostate Cancer UK. All the work I've told you about was done by a graduate student in my lab, Andrew Best, who's currently writing his thesis. I'd also like to thank Alison Tyson-Kappa, who jointly supervised this project, and Thomas Kirk, who helped us with the iCLIP bioinformatics, Yone Uhl and Julian Connie gave us a lot of advice, gave Andrew in particular a lot of advice by very quickly responding to emails about questions about setting up iCLIP in our lab. Thank you.